Matthias. This is my senior biological sciences A project. I used oleic acid, which is a specific inhibitor of conjugation. It stops, it binds to the ATPase, through which bacteria form the syringe to inject other bacteria with the genes for antibiotic resistance and conjugation. So I applied this in a couple different scenarios. One was in a smaller population of antibiotic resistance mixed with a larger population of non-resistant bacteria. So what I found was that in this scenario, if you applied oleic acid and antibiotics at the same time, you could decrease the total amount of antibiotic resistance genes in the population compared to just applying the antibiotic. The other scenario I did was two different populations, two different resistances of different drugs, and you could apply them, you basically check the transconjugate population that was resistant to both of the drugs. So what I found was that one, they were very willing to transconjugate even in the absence of an antibiotic pressure, and the other one was that you could apply oleic acid to reduce the total amount of transconjugates. So the interesting clinical or potential application here would be multiple drugs being used at the same time on a patient to try and eliminate a nasty strain. You've got sort of worries of the confounding factors from the gut microbiome. It's very gene dense. There's a potential for the transfer from this dense bacteria space to these other bacteria and the formation of a multi-drug resistant strain or a superbug or something like that. So you could sort of prevent that or slow that process through the application of these drugs, inhibitors of conjugation, or you could apply them preventatively in conjunction with antibiotics and sort of have the antibiotic applying selective pressure, but at the same time you have this oleic acid stopping the spread of antibiotic resistance within the population if it does arise due to the selective pressure exerted. I got my idea more or less just by journal trawling. What I thought was funny was that, so I would start at this one thing, I was like, oh, this is an anticonjugate, and then I found another paper which was like, that's non-specific, that doesn't work. And I went through like 4 different iterations of that.